My name is Joshua Gann, and I'm a research tech in the Tretyakova Lab. And today I'll be teaching you how to use the Mermaid 4 DNA synthesizer. To start off, we'll be reconstituting and adding new phosphoramidite to the instrument to replace old ones. You should be doing this about every week or so to ensure that your phosphoramidite is as fresh as possible. So to begin, we'll take our old or our new phosphoramidite and we will reconstitute it in dry acetonitrile. To do this, we've evacuated this headspace of a dry acetonitrile bottle with argon, and we will now draw up 10 mils for every half gram of phosphoramidite. So once we have our desired volume, we're going to invert the syringe and then push out any air bubbles that may be there. We don't want any spraying to occur. Now that we have our desired volume, we're going to crack the lid on the phosphoramidite bottle and spray the dry acetonitrile along the sides of the bottle. This ensures that there's no splashing occurring and that we're not introducing any moisture to the phosphoramidite. This is really critical because phosphoramidites degrade under moisture. So we're spraying along the bottle and slowly spinning it to get all the sides. And once we have all the phosphoramidite down to the bottom, we're just gonna squeeze out and quickly cap once all of our acetonitrile is out. We now need to reconstitute this and make sure that all the phosphoramidite is completely dissolved. So what we'll do is we'll just vigorously stir here on the tabletop. Now that we have a nice and clear solution of phosphoramidite, we'll now put it onto the instrument. To do this, we'll first remove our old phosphoramidite. We will then quickly open, insert the hose, and screw on to the left, making sure that it's finger tight, but it's not too hard to get off later. Now that we have our new phosphoramidite on the Mermaid, we need to purge it in. So to do this, we're gonna enter into the Poseidon software. So we'll click the icon here that says Poseidon, and we'll log in using the username of admin and the password admin. We'll then go to valve options and injection head test. From here, you can see all the various reagents that are on the Mermaid. And when you're getting ready for a synthesis, you should be purging all of these into the lines. For now, we're gonna focus on the phosphoramidite that we've added to the synthesizer, DA. So to navigate to it, you'll just click on the icon right above A, and we'll then inject 100 microliters at a time of DA solution. What we should be seeing when we're observing through the porthole is a nice steady stream that shows that the reagent is all the way up to the injection head. You can notice that there's no bubbles occurring in this stream, and it's very straight and quick. So now that we're ready to set up our run, we need to write a sequence file for it. To do this, we'll go into File Explorer, click Sequence Files, and copy a previous file. With this copy, we'll rename it to have the dates, a gene of interest, the initials of whoever's synthesizing it, and any other naming that you want to include for your sake. With this file, we can now write in whatever we want past the comma. For DNA, you'll want to write in uppercase, i.e. A, T, C, and G. And for RNA, you'll want to write in lowercase, so A, U, C, G. This will tell the instrument what basses you're trying to couple to your column, and will allow you to designate where in your sequence these are going to occur. Now that we have this, we'll save it and close out. Now that we're ready to set up our run, we're going to install our column onto the synthesizer. To do this, we'll take a CPG column that contains 1 micromole of resin and is coupled to the last base of your sequence. We will open up the synthesizer door and install the column onto one of the 4 positions within your synthesizer. We will now close our doors and lock it in place. Now that we have all of our reagents in place, we can now set up our run. To do this, on the main screen, we'll click Set Up Run. We'll then select a sequence file for the position that our column is in, using a sequence file that we made earlier. We will then switch over our column to a standard column by either selecting Set All Standard or clicking individually on the base. With this, we need to change to start at base 2 because we already have a nucleotide attached on the resin. Select Next and select a script file. The general script file that we use is this modified bio 1 micromole standard column XY coupling LGC from November 2021. We'll then select Next. On this screen, we see an estimated usage in terms of volume for all of our reagents. Make sure that all of your reagents have enough volume to continue with your synthesis before starting. The lot information tells you where each of these reagents come from. However, we do not use this very often. You'll then want to label your run name, so we'll label this test run. And then you can add some notes to say what this strand is going to be used for, what it contains, and what different modifications are on it. You'll then want to run an argon high and low flow test. This makes sure that your system is not being overpressurized or underpressurized. Once you have these 2 green lights here, you can proceed. Here we have another injection head test. You'll want to make sure that all of your reagents are purged in like we did earlier. We will then wash the column twice by clicking number of steps up here to be 2, and then we'll click Wash. If you have multiple columns, you can also select Wash All. The synthesizer will then run acetonitrile over your column to rinse out the CPG resin. The final screen will ask you if you want to start your run, and if you are very sure, you can select OK. Now the inner chamber of the mermaid will start to purge with argon, making sure to keep everything under an inert atmosphere. So now that we're at the run screen, we can navigate to a couple of different places during our synthesis. At the moment, it's paused, but while it's running, you can look at your block info and see exactly what step it's on. You can also navigate to your Triddle graph, which will tell you what protecting groups have been removed at which bases. At the moment, there's nothing right now because we're just starting our synthesis. You can also see on the side here the steps in order of their occurrence. And they're organized by cycles and the bases that they correspond to. From this screen, you can also choose to pause at particular steps. So if we were to right-click, we can say pause here. We can also reposition our step by doing the same thing and right-clicking and clicking reposition. This will move around the current status of our cycle to a place where we would like it to be. In this case, I'm gonna keep it on the wash. So now what we want to observe is at the start of our run, we want to be able to see an obvious deprotection of our base from the column. We want to make sure that those trittle groups are coming off so that incoming phosphoramidite can couple onto the column. We're going to go through a couple of wash steps here first, and then we'll see the D-block reagent enter into the column and cause an obvious orange color to appear. You should ideally see this at every step of your run when you're D-blocking. So now our D-blocking reagent is going to enter the column. It's going to flow through, and you can see that there's that nice dreamsicle orange color there. As you're doing this, you should monitor to make sure that the instrument is not stopping and that there are no errors occurring. After your D-block step, your synthesis will run through a couple of different steps, including washing, coupling, capping, oxidization, and the later deprotections. As you're going through this, you should monitor your reactions to make sure that there's trittle data for them and that there are no errors occurring during this process. To monitor your trittle data, you can go to the block 1 trittle graph and refresh data. And as we can see, there's a nice bar graph that's being formed as you're going through your synthesis. Make sure that this signal is appearing for each one of your bases. The intensity of these bars is not relevant to how efficient they're coupling, but it is a good indicator that your reaction is working. Ideally, we want to see a signal for each of our bases. If we don't, we want to try to alleviate that through some troubleshooting. Now that our synthesis has concluded, we want to deprotect our oligonucleotide from the column. To do this, we'll remove our now dry resin column from the synthesizer. We will take a razor blade and cut just above the frit, making a sawing motion just above. This will remove the excess plastic and leave the frit open to us. Now, using a syringe needle, we're going to remove the frit. So you'll just plunge it into the frit and slowly pry out. You'll eventually see it come loose, and then we can remove it with our hands, or not. There we go. So now we've exposed the resin, and we can place it into our microcentrifuge tube by inverting and then tapping loose. We'll tap loose all of our resin, and then we will add ammonium hydroxide to deprotect. Modified bases will require different types of deprotections, but generally for our syntheses of normal oligonucleotides, we will use ammonium hydroxide. So we're going to take our syringe here, we're going to add 1 ml of ammonium hydroxide. Right. So now we'll just very quickly agitate our resin so that it's suspended in solution, and we'll add it to our inverter. This will now deprotect overnight for at least 17 hours at a maximum of 24. With our resin now on the inverter, it will go through the process of deprotection for at least 17 hours and up to 24 to allow the DNA oligonucleotide to be removed from the resin and fall into solution. As you're going through cleanup of the Mermaid synthesizer, you want to make sure that you're disposing of things properly. So any needles that you might have used during your reaction, you want to dispose of those in sharps containers. Any garbage you want to throw into the trash. And then any empty jars that you may have, you want to rinse them thoroughly with acetonitrile and then dispose of them in glass waste and putting the reagents into our liquid waste. Once you have that done, you can allow the instrument to set. To keep the instrument in good maintenance, you want to make sure that you're cleaning the lines as well. So to do this, you'll just flush it out with any excess reagent you may have in your system to help purge the lines and keep any crystals from forming on the injection head. This will allow for other syntheses to work well and for the instrument to be in good maintenance. As you're going through oligonucleotide synthesis, you may need to use specialized phosphoramidites in order to introduce certain epigenetic bases or modifications to your DNA. When you're doing this, always refer to the manufacturer protocols and the protocols for the DNA synthesizer so that you can put these specialized phosphoramidites in small aliquots and inject them onto the instrument properly. If you experience any technical difficulties or have any questions about DNA synthesis, you can contact LGC Biosearch Technologies at 972-335-2525 for more information on the Mermaid 4 synthesizer. If you have any questions about reagents and other phosphoramidite materials, you can contact Glenn Research at glenresearch.com for more information on the different reagents that are compatible with the Mermaid 4 synthesizer. If you have any immediate questions or are going through some trouble with the DNA synthesizer, contact a lab manager or the lab tech that's responsible for the Mermaid.